Thanks, Jim. Thanks for that kind introduction. Today what I'd like to do is talk in general terms about the classification and monitoring of hematopoietic neoplasia in the genetic era. And by necessity, we'll emphasize the contributions that our laboratory has made to this general topic. And I apologize in advance, we're going to go kind of fast through a lot of relatively detailed information. So, to start with, a little bit of background. This is me in 1978, when I was in high school, when I had hair. Same person. So, the basic principles of flow cytometry are pretty much the same regardless of whether one's doing diagnosis, classification, or monitoring for disease. And it revolves around the expression of proteins or antigens on the cell surface. And the reason this technology works is that, as it turns out, normal cell populations express these antigens in a reproducible and consistent way with maturation along a given lineage at a particular maturational stage. And when cells become neoplastic, they acquire a series of genetic mutations that we'll talk about in more detail that result in changes in these patterns of protein expression, which can take a variety of forms: increases or decreases in expression, abnormalities in the timing of expression, homogeneity of expression. But in some way, the neoplastic population differs from the population of cells of similar type that you would find in a normal tissue. And we rely on those differences to allow us both to identify the population, then to classify it, and finally to monitor it after therapy. So what this implies is that one can create maturational maps, in this case of B-cell maturation, where one can look at a variety of different antigens associated with B-cell differentiation and identify the stages along that differentiation pathway where these antigens appear and disappear, in which they're expressed, and create maturational maps of this sort. What's important about these maps is that with each particular stage of differentiation or maturation, there's a unique immunophenotype that's characteristic of that stage of differentiation. And it's that composite immunophenotype that we use to identify and categorize these cell types. These types of maps can then be directly translated into reagent panels of antibodies, in the case of flow cytometry, and used to identify discrete cellular subpopulations. So this is an 8-color flow cytometric experiment that was done more than 10 years ago. We were the first laboratory to use 9 and 10 color flow in the clinical laboratory environment. And it demonstrates the high degree of consistency in the expression of many of these proteins. The expression of these proteins is not at all random. It's very highly correlated. And as a result, you get these discrete groups of cells or events, with each dot here representing a single cell, characteristic of specific stages of maturation along the B-cell maturational pathway in this case. One can then take this basic immunophenotypic information and apply it to clinical samples and identify situations where the immunophenotype is different from what we see normally. In this case, an example of an immature B-cell neoplasm, precursor B-cell lymphoblastic leukemia, where the population in red represents the abnormal population. It's both markedly expanded, expresses many of the antigens typical of B-cell differentiation, but the intensities of expression are now different from what we see during normal maturation. So for instance, CD45 expression here is essentially absent on the population of interest, whereas it should be expressed at a low level normally on immature B cells. CD19, although expressed, is expressed at an abnormally low level. CD10 is expressed at an abnormally high level. There's a little bit of expression of CD13 and 33, two myeloid-associated antigens, which is aberrant. And CD38 expression is markedly decreased to almost absent, which is very abnormal for a normal immature B-cell population, where the level of expression is more at an intermediate level of intensity. So by looking at the composite immunophenotype for a particular population of cells, one can make the correspondence between that immunophenotype and its normal counterpart within the maturational sequence. That information is then used both to identify the fact that the population is aberrant, and then also used to classify the population along the maturational scheme, which is how most of the current classification systems are at least in part based. So if one looks at the immunophenotype of B-ALL, the disease that we were just looking at, it's the main type of B-cell acute leukemia, we can do that kind of association and create a variety of different classification subgroups based on their similarity to these normal stages of maturation. And this is what has been done historically. The problem with this type of an approach, as it turns out, is that these subtypes have essentially very little association with outcome, particularly if one takes into account various genetic factors that we'll discuss a little bit later in the talk. So immunophenotyping, although it's very descriptive and useful for identifying aberrancy, doesn't necessarily correlate with treatment response or outcome. It's not independent of genetic factors, and it's in fact the genetic factors that contain most of the information that's associated with outcome. And as a result, it's relatively not used anymore. There was a time back when I was in training where this was of much more interest, but now it's recognized that genetic factors really are pretty much what drive classification now, and these immunophenotypic classification schemes are essentially no longer used in clinical practice. So when one then goes to the genetic level and starts asking the question about what the degree of heterogeneity is within this particular, otherwise seemingly relatively uniform disease subtype, one finds that there is actually a lot of heterogeneity at the genetic level underlying this disease. So in this particular experiment, this group looked at the gene expression profile of a series of B-ALL patients where they excluded many of the known molecular subtypes of B-ALL, such as those containing the 1221 translocation, trisomies of chromosomes 4 and 10, et cetera. And what you're left with are 8 or 9 different unique genetic signatures within the B-ALL subset, only 2 of which had abnormalities that had been previously described, the MLL abnormalities and translocations involving chromosomes 1 and 19, suggesting that there is heterogeneity, but yet there are discrete different genetic subgroups within B-ALL beyond even what we currently understand to exist. In particular, one of the subgroups that was recently identified by a couple of different groups, including the Children's Oncology group, is a group of B lineage ALL that has a gene expression signature that's very much like what is seen in patients that have the Philadelphia chromosome positive form of B lineage ALL, and so have been termed PH-like B lineage ALL. And these are identified down here on the bottom left as a unique kind of subgroup. Part of the reason for interest in this particular subgroup is that it's been recognized that this subgroup is associated with an inferior outcome. So if one compares B lineage ALL of other types, here they have a relatively good outcome with modern therapy, whereas BCR-ABL positive B-ALL has a relatively poor outcome, now somewhat improved since these studies as a result of use of tyrosine kinase inhibitors. And the BCR-ABL-like subset has essentially an identical outcome to that of the patients that have the frank BCR-ABL rearrangement. So there seems to be a similarity both at the gene expression level as well as in terms of clinical outcome for this particular disease subset. Since we know that the BCR-ABL positive B-ALL responds to tyrosine kinase inhibitors, the question now is, do these BCR-ABL-like forms of B-ALL also respond to tyrosine kinase inhibitors? And that's something of active investigation going on within a couple of the different disease groups, particularly the Children's Oncology group. More detailed genetic examination of this particular subset has revealed that the result or the cause of these underlying gene expression signatures are the presence of novel tyrosine kinase fusions. And this has been identified by transcriptome sequencing. So a certain subset of these involve the ABL1 locus, similar to what we see in Philadelphia chromosome, but they have different partners. Rather than BCR, we have a variety of other genes that are involved in partnership in a unique fusion with the ABL1 mutation or gene. Similarly, JAK, another tyrosine kinase, has a novel series of fusion partners. PDGF receptor beta has a novel fusion partner, and there's an ever-growing list of these particular abnormalities that are seen in this subset of B-ALL. These cases constitute about 15% of pediatric B lineage ALL, and the presence of mutations involving the CRLF2 gene, which we'll talk about in detail in just a minute, account for about 50% of this particular subgroup. So again, part of the reason these are of such interest currently in the current environment is that there's the possibility that this very poor outcome subset of B-ALL might be positively affected by the use of tyrosine kinase or other inhibitors targeted against these types of molecular lesions. The CRLF2 gene itself, which has been of focused interest of the group in which we've participated in a variety of studies, stands for the cytokine receptor-like factor 2. This is a protein that forms a heterodimer with the interleukin-7 receptor, or an important growth factor receptor during lymphoid development, and is in fact the receptor for the thymic stromal lymphopoietin. There are 2 mutations that are described as the major mutations in CRLF2. One is a deletion of a gene called PAR1 on the X chromosome, yielding this particular fusion protein or construct. The other is a rearrangement that juxtaposes the immunoglobulin heavy chain enhancer next to the CRLF2 gene. In both cases, there's an upregulation of expression of this particular protein on the cell surface, and hence an increased signaling that then drives cell proliferation. This particular mutation is also associated with other mutations, such as mutations in the JAK gene, JAK1 and JAK2 in particular. These are seen in about 60% of patients that have the CRLF2 mutation, as are mutations in Icaros, IKZF1. So there's a strong association between these three genes and this particular subset of ALL. If one looks at the expression of the CRLF2 gene itself, one can see that it's in fact these cases that have CRLF2 disruptions, translocations, or mutations that are driving the increased overexpression in this particular subset. CRLF2 mutations in particular are about 7% of B lineage ALL, but are 50% of patients with Down syndrome, so very high representation in that particular subset. And there's a very strong ethnic association with Latino or Hispanic ethnicity as well. So we see a lot more of these in the US than you do in Europe, for instance. One of the studies that we've been involved with is looking at a comparison between the expression of the protein as measured by flow cytometry in comparison to both FISH and PCR-based techniques. And as it turns out, flow cytometry is very nicely able to distinguish that subset of patients that have both or either one of these two genetic abnormalities involving the CRLF2 gene. These would be the negative samples down here. These are the samples overexpressing CRLF2 on the cell surface. In comparison to PCR-based methodologies, flow cytometry almost dichotomously categorizes a subset of positive cells that are the ones that have these underlying genetic abnormalities and negative subsets of patients that don't have these abnormalities. In contrast to PCR, which shows a much more continuous distribution, the high level expression paralleling the high level of protein expression seen by flow cytometry, but also showing an intermediate level of expression on a large number of cases that flow cytometry does not show increased expression of protein. As it turns out, we believe that this subset of patients have this overexpression due to a gene dosage effect, which is due to the presence of trisomy X. Remember, CRLF2 is on the X chromosome, so the presence of extra copies of X is giving rise to increased transcripts that aren't necessarily converted into increased protein on the cell surface. So flow cytometry appears to be a very excellent screening tool for looking for this particular subtype of PH-like B lineage ALL. In addition, we've shown that this disease can be subclonal, in that you have patients with B lineage ALL that may not show whole-scale overexpression on the entire population, but contain small subpopulations of cells overexpressing CRLF2, as is shown on the left in 2 cases. We've sorted these populations and performed FISH and confirmed that these small subpopulations, unlike the bulk population, have deletions of the CRLF2 gene consistent with the genetic abnormality underlying that, as well as specific fusion of CRLF2 with this other marker gene on chromosome X. So there appears to be the acquisition of CRLF2 mutations in patients with B lineage ALL as a secondary type of abnormality. And this work is in preparation for publication. So if we look at the genetic classification of B lineage ALL, we have a subset of mutations that have been identified as being good risk, indicated here, and a larger subset of mutations that are considered poor risk, indicated here. And this kind of genetic risk stratification is what's now used commonly in clinical trials of B lineage ALL to stratify patients to different treatment arms and to drive therapy, in contrast to immunophenotypic types of classification. B lineage ALL has been relatively well studied, as you can see, by both flow cytometric and molecular techniques. T lineage ALL has been less well studied, in part because of its reduced frequency in the general population. Nevertheless, there have been immunophenotypic classification schemes that have been proposed for TALL. This is probably the most widely used one from the EGIL group, dividing TALL into relatively more immature subsets on the left, more intermediate stage of differentiation called common thymocyte TALL in the center, and more mature subsets of TALL on the far right. And there are immunophenotypic differences that you can see that help to categorize these patients into these different subsets. This is an example of a case of TALL showing expression of T-cell associated antigens like CDs2, 5, and 7, but importantly, co-expression of both CD4 and 8, and co-expression of CD1A, which are the hallmark of the common thymocyte subset of TALL. so one can use this immunophenotypic classification to assign the patient to that particular subset of disease. There's very little data in terms of the relationship between immunophenotype and outcome for TALL, but this study recently published from the European group does demonstrate that in fact patients with the cortical or common thymocyte subset of TALL have a somewhat improved outcome in comparison to those with the more immature or early or later stage or more mature subsets of TALL in this particular study. It's important to remember that outcomes of this sort are highly correlated with the type of therapy that's used for these particular subsets of disease. About the time that these data were being compiled, the group at St. Jude's, under Dario Campana's direction, identified a discrete subset of TALL with a particularly poor outcome that they called early thymocyte precursor, or ETP TALL, thought to be one of the earliest immunophenotypic subsets of normal T-cell development, the early thymic precursor. This type of TALL was immunophenotypically defined and identified principally by three features, one of which is the expression of a variety of either immature or myeloid-associated antigens, indicated here in the bottom row, which this case shows positivity for at least two. The absence of expression of somewhat later stage antigens during T-cell development, CD1A and CD8, which this case lacks, and the low level of expression of an antigen CD5 that appears later during T-cell development. And the criteria for assessing CD5 expression are a little bit arbitrary, being less than 75% positivity or a level of expression greater than 1 log lower than that seen in normal mature T cells. So since our laboratory now serves as the single reference laboratory for the Children's Oncology Group for TALL MRD detection, we were asked to evaluate for ETP expression in TALL to associate with outcome in these large clinical trials. So we correlated the assay that we were using with Dario Campana's assay, reviewing cases in parallel to make sure we were employing his criteria similarly, and during that process recognized that in addition to these early thymic precursor, or ETP subsets of cases, there's another subset of cases that we called near ETP that met all of these criteria except they had a higher level of CD5 expression. We didn't know what the significance of that was, but it was just operationally convenient to put them in this category so we could keep track of them while we were doing this analysis. We are also, as a result, part of a large consortium that did whole genome sequencing of ETP TALL, recently published in Nature, where there was an attempt made to identify the underlying genetic mutations in this subtype of TALL. And as one can see, essentially there was no single underlying genetic abnormality that was identified. Instead, this is a very heterogeneous type of underlying mutational profile with many genes mutated being in signaling pathways, developmental pathways, or chromatin remodeling pathways, but no single underlying genetic lesion that could be used to define this particular disease. So the definition of the disease of this subtype remains primarily immunophenotypic at this point. This was then employed on a very large clinical trial, and the first results of this trial I presented at the American Society of Hematology meeting this December as one of the 6 plenary presentations for that meeting. We enrolled 1,895 patients with TALL on this trial over a 7-year time frame. 1,144 of these had complete data regarding ETP status and residual disease monitoring, or MRD. We assessed the ETP routinely at diagnosis but didn't report it to the institution, and then assessed MRD at a few different time points after therapy. What we were able to demonstrate is that the frequency of ETP in this very large cohort was similar to what Dario had described in the original St. Jude's cohort, being around 11 to 12%. The near ETP subset actually is slightly larger than that. This is a novel finding that hadn't been reported to date, and those that had other subtypes form the majority of TALL. We were able to show that patients with ETP, similar to what Dario had shown, have a relatively high incidence of MRD that's present at the time of day 29 after the end of induction therapy. Here we're reporting that as the number of patients that were MRD negative, which is relatively rare. These patients are also associated with induction failure at a higher rate. Near ETP also shows a higher rate of MRD positivity at the end of induction and a similar rate of induction failure, suggesting that biologically, at least under this type of therapy, these patients behave very similarly, in contrast to the not ETP patients that have less frequent MRD detected and also have a much lower instance of induction failure. The thing that's striking about this study, though, is that despite these differences in MRD detection, residual disease after therapy, and induction failure, the actual outcomes for all of these groups was the same. So if one puts this in context, on the right-hand side, we have the data from St. Jude's where they originally described ETP. And notice the extremely poor outcome that ETP has in their particular study. On the left-hand side is the data from the trial that we just completed. There is no difference in outcome for ETP whatsoever, or near ETP. They all look the same. So the changes in therapy that were made as part of this trial with the Children's Oncology Group have essentially eradicated the bad prognosis outcome that this particular subset of disease appears to have. And this is a novel finding and why it was a platform presentation. Notice also that all the events that are occurring happen within the first 1 to 2 years after therapy, and after that the patients essentially remain same without relapse after that. So it's an early process by which induction is induced. There are other classification schemes out there other than immunophenotypic for T-ALL, and this is one example using gene expression array data, not unlike what I showed you for B lineage ALL, and it can very nicely divide patients with T-ALL into at least a few different subgroups, one of which is the immature subset, which is where ETP lies. But the application of this in a clinical setting or a clinical trial setting has yet to be demonstrated, and so is something for future work. AML is an even more complex problem. The disease is very heterogeneous, and as a result, classification is also very heterogeneous. But at the genetic level, there's been a lot of progress made over the last few years, recognizing, first of all, that AML has a much lower mutational burden than solid tumors in particular, where their mutational rates are much more frequent in terms of the number of genes. And also in identifying the genes that have been significantly mutated as a result of the Cancer Genome Atlas project. 23 genes have been shown to be recurrently mutated in AML. Most of the high-frequency genes are ones that we already knew about prior to the project that have been identified using various other means, but there's this long tail that extends out to hundreds of genes. In the initial publication, 237 additional genes were mutated. This is probably an underestimation in terms of the way that data was analyzed, and it probably is many hundreds of genes that are actually mutated, such that every case of AML is probably somewhat unique in terms of its overall mutational profile. These can be demonstrated in this kind of a plot where one can see the categories of AML that we recognize by recurrent genetic mutations such as acute promyelocytic leukemia, inversion 16, 821 AML, and NPM1 mutated AML, which forms a large subset of particularly the normal karyotype subset of AML. But notice the large degree of other associated mutations that occur as part of this, as well as the number of other subtypes of AML that are relatively low in frequency but have relatively unique recurrent types of genetic abnormalities. So AML is, at the genetic level, a very heterogeneous disease, probably more so than what we see in B and T lineage ALL, and hence classification is increasingly problematic. So what we can say in terms of classification overall is that immunophenotypic classification uses the closest normal counterpart as the way to structure or classify these subsets of disease, but it lacks specificity in terms of clinical outcome, and the correlation with clinical outcome is at best limited, and most of the information really comes from the underlying genetic lesions. So increasingly, immunophenotypic subclassification people are moving away from. Genetic classification uses recurrent genetic abnormalities to do classification, with the idea that recurrent abnormalities are those of increasing importance that probably are something biologically important to the patient's leukemia, but they reveal profound genetic heterogeneity underneath this, such that in many cases every tumor may be in fact genetically unique, and so presents a real challenge to classification and therapeutic schemes. So that's kind of classification of acute leukemia in a nutshell, where we currently stand. Let's talk a little bit more about the monitoring of patients' disease after therapy, which is an area that our laboratory has had a lot of involvement over the years. One uses, by flow cytometry, exactly the same principles that we initially outlined to do monitoring. We're looking for differences in immunophenotype for immature populations of cells that are different from normal patterns of maturation. Here we have an example of B lineage ALL, where normal maturation is indicated by the cyan or light blue population here. These are normal B cell precursors. Mature B cells are in dark blue here. The population in red represents residual disease for this particular patient, where the level of CD10 and its co-expression with CD20 are different than any normal stage of B cell development. CD58 and adhesion molecules overexpressed. CD45 has been downregulated, etc. So one can, in most cases with B lineage ALL, define immunophenotypes that are distinct and unique for a patient's leukemia that are different from normal and use that to monitor disease. Here at a level of about 0.1%, which isn't all that low for these types of studies now. This type of evaluation looking for residual disease has been shown to be highly significant in terms of clinical outcome. And so data from the Children's Oncology Group shows that patients with increasing levels of residual disease at the end of induction therapy have increasingly poor outcomes, such as the patients with the highest level have the worst outcomes. And these are in very large cohorts consisting of thousands of patients, so the statistical significance is very high. If one uses multivariate models to look at the relative importance of various prognostic factors, it turns out that the measurement of MRD at the end of induction at a level more than 1 cell in 10,000 is the most significant in terms of outcome for patients with B lineage ALL. More so than age and white count, the standard NCI risk groups, more so than many other genetic abnormalities. And increasingly, people are beginning to believe that while genetic abnormalities are useful for risk stratification for initial therapy, in fact, the eventual outcome is more strongly associated with the actual response of the patient to a given therapeutic protocol. And so the measurement of disease after therapy is increasingly becoming important to clinical oncology groups. In TALL, the data is much more limited, and we're really the only group that's published any large-scale study of TALL. This publication is in press, or in the works. And what we're able to show is that in patients with TALL, those that don't have the ETP signature, MRD is able to stratify patients in terms of outcome. Patients that have low or almost undetectable levels of MRD do much better than patients with higher levels of MRD, and patients with relatively high levels of MRD have the worst outcome, as you might expect. However, in the ETP subset of patients, most of these differences are not present unless you have extremely high levels of disease. Again, suggesting that the therapy used in this particular trial was unusually effective in ETP patients. These are the really worst outcome subsets with high levels of residual disease after therapy that it can be the target for novel therapeutic intervention. And that's what the current generation of trial is focusing on. If one goes further out from end of induction therapy and looks at later time points, such as the end of consolidation, one can show that the presence of MRD detectable at even lower levels, such as 0.1% in TALL, is associated with a worse outcome in comparison to patients that have lower levels of MRD. And so it may be that in TALL, because of the slower kinetics of response compared to B lineage ALL, that we need to be looking at later time points from therapy, not at very early time points like day 8 in the peripheral blood or day 29 after induction chemotherapy. And this is the subject of investigation on the current clinical trial as well. A few groups have also looked at flow cytometric evaluation of residual disease in patients with AML. Our particular area of interest has been in transplant patients because of the quality of the transplant data we have available at our institution. And this shows one of the multiple studies that we've now published in AML showing that if one detects the presence of residual disease prior to the patient going into bone marrow transplantation, those patients that are positive for disease at a level that's not morphologically evident have a much worse outcome than patients where disease is undetectable. And it doesn't matter whether the patient's in first or second clinical remission going into transplant, the outcomes are essentially the same, with a hazard ratio for relapse of about 4.9, which is very high and highly statistically significant. Unlike in B lineage ALL, and to some extent T lineage ALL, the level of MRD is more controversial. In our data set, the level of MRD that was detected going into transplant was not really strongly associated with outcome. It really was just the absence of MRD that predicted a good outcome or a good risk subset of patients. There may be a variety of reasons for this, part of them sample related due to hemodilution of the samples that we receive, and part of it related perhaps to the way in which the populations are quantitated, but nevertheless, this has been seen in our group and also by at least one other group in the published literature. So it appears that the monitoring of MRD by flow cytometry, and there's also similar data using PCR-based techniques for certain subsets of these diseases, is extremely useful in terms of determining or predicting the patient's outcome after therapy. The question with flow cytometry has more to do not with whether it's effective in terms of identifying patients that have better or worse outcomes, but is it reproducible? And partly this question arises because the application of this technology in the clinical context is highly heterogeneous. Different clinical laboratories use different panels of reagents, evaluate data in different ways, and have different levels of experience with this technology in doing this type of work. And so there's a lot of skepticism about the ability of flow cytometry to be reproducible in terms of the detection of residual disease. So we've done some work looking at this particular issue within the context of B lineage ALL MRD detection. The particular assay that we're using has two different reagent combinations, here labeled as tube 1 and tube 2. And if you use those independently from each other, you can show that on the same samples, either of those combinations performs extremely well and is highly reproducible in quantitating the level of residual disease within a single assay. More importantly, one can look at assays between two expert laboratories, our laboratory here and Johns Hopkins University Laboratory, these being the two reference laboratories for B lineage ALL MRD detection in the Children's Oncology group, and show that there is essentially the same frequency of detection of MRD positivity at different time points after therapy. One can also show that the level of MRD that's detected between the two laboratories at different levels of MRD detection is essentially the same between the two laboratories. And more importantly, one can look at outcome data and show that, in this case, for a small subset of patients that were an intervention group on this trial, where we were intervening and giving additional therapy if they were positive above the level of 0.1%, that we again see the exact same types of outcome for patients between the two laboratories that are divided between the East and West Coast geographical regions. And in particular, this particular survival curve has a very unusual shape to it. This intervention appeared to have delayed the onset of the poor outcome that this patient's subset would normally have, in that in the first 2 years after therapy, the outcomes appeared to be essentially identical to what we would expect to see in good risk patients, but after about 2 to 3 years, they deteriorated and assumed the poor outcome that had been seen in prior trials. So this particular therapy was effective in initially treating the patients into remission, but not in terms of long-term outcome, and so different types of therapeutic strategies are needed. But the point here is that one can actually show essentially the exact same data in terms of assay performance between two expert reference laboratories, even to the extent of showing similar outcomes in large clinical trial settings. So flow cytometry itself is capable of reproducible use in a clinical trial context for residual disease monitoring. It's not something inherent to the technology. The challenges with reproducibility tend to be more in terms of politics between laboratories and the willingness of the community to employ the same techniques. So what we can say about flow cytometry in this context for monitoring is that it's fast, it's relatively inexpensive, there's a large instrument base of this type of equipment out there in the community, so in principle this could be relatively rapidly translated into other laboratories, and it can be made reproducible provided one agrees to use the same protocols, which has been the obstacle to this point. However, the disadvantages of this technique are that it requires somewhat subjective interpretation. As you've seen, we're looking at graphical dot plots and looking at groups of events that are distinguished from normal, and this requires some subjectivity and experience on the part of the interpreter. There also are changes in immunophenotype that occur after therapy that can make this more challenging to employ. The sensitivity of the technique at the moment, I would say, is of moderate sensitivity. It's not as highly sensitive as some molecular techniques now can provide, and the standardization at the moment is really quite poor. So if we were to employ this, we would need to undergo a major standardization type of effort nationally in order to make this technology widely available. In part for these reasons, we began to look at other ways of determining residual disease after therapy, and so became interested in next-generation sequencing as a way to measure residual disease after therapy. And the principle here is relatively simple, in that we can determine a unique sequence characteristic of the tumor at diagnosis, we can deep sequence that locus in follow-up samples, and then simply count the number of these unique sequences that occur after therapy. Sounds pretty simple in principle. In the case of lymphoid neoplasms like B or T ALL, we have polymorphic loci, the immunoglobulin heavy chain receptor, the T cell receptor genes gamma and beta, that provide relatively unique sequences that are essentially tumor specific because of the way these genes reorganize during development. We also have genes that are associated with leukemia where there are smaller point mutations or small insertions or deletions that could potentially be targets for a similar type of approach as tumor-specific markers. So the question more recently has been demonstrating whether this is in fact feasible as a general strategy. So as I mentioned, the immunoglobulin heavy chain and T-cell receptor genes recombine during development to form a construct that has a high degree of diversity related to these variable regions, the N regions, in the overall sequence that serve essentially as a tumor-specific marker because of their low frequency in normal populations. So one can develop assays against these areas, in this case TALL, to demonstrate how this might work. And we were in fact the first group in the world to publish evidence about using next-generation sequencing for MRD detection. In this case, the example was TALL. So here we have a cohort of TALL samples where we have used sequences against the T-cell receptor beta chain, have amplified across this highly diverse region, used this as a tumor-specific marker, and compared it with flow cytometry. And one can see that the majority of patients with TALL on the top are informative in terms of having a unique sequence that can be identified by this technique, but there are a subset of patients that lack this unique sequence, and these turn out to be the immature subset of TALL of either ETP or near ETP type, as we talked about previously. So it's not uniformly informative using the beta chain locus for this assay. But in those cases where it is informative, there's a good correlation between flow cytometry and the quantitation of residual disease on a subset of samples, as indicated here, that were positive by both techniques, on a subset of samples that were negative by both techniques, and there's discordance on a subset of samples where next-generation sequencing is finding lower levels of residual disease than flow cytometry in this particular assay was designed to detect. And so there are these discordant samples, and the outcome for these patients has yet to be elucidated, so we don't know what the actual clinical impact is. But it nevertheless shows in principle how one can use next-generation sequencing as a monitoring tool in TALL. One can do the same thing not using the beta chain gene, but the gamma chain gene of the T-cell receptor. In this case, the gamma chain gene is rearranged on both alleles in about 78% of the cases, and one can show a high degree of concordance in the rearrangement of these two alleles in terms of MRD quantitation, and show very similar high correlation between flow cytometry and next-generation sequencing for positive cases, negative cases, in this intermediate subset that has low-level positivity by sequencing, but an absence of detection by flow cytometry. Again, the clinical significance is unclear, and this is the subject of an R01 grant that we have to look at a larger cohort of these patients in real time on the current ongoing trial in TALL. The same thing can be done in B lineage ALL, and so our group, as well as one other group, have shown the feasibility of doing this, where in this good risk cohort of B ALL patients from the Children's Oncology group, we again show good correlation with flow cytometry on those cases that were positive by both, cases that are negative by both, and this intermediate subset where next-generation sequencing finds residual disease that's not seen by flow cytometry. Note the subset of cases that actually have a level of disease that's in the range that flow cytometry should be able to detect. And I can assure you I've looked very hard at this subset of cases, and there isn't anything there that you can pick up immunophenotypically. So we did some sorting experiments and demonstrated that at least in some cases we think that these are due to maturation of the leukemic population to a mature subset that's immunophenotypically indistinguishable from normal mature B cells. The other cases may be due to degraded cells that result in DNA that's available for amplification but not detectable by flow cytometry. The exact explanations still have to be worked out. But it's not a unique observation just to next-generation sequencing. It's also seen in standard PCR data, such as is often collected in Europe, and that was published about a year ago. What about leukemias like AML, where we don't have nice polymorphic loci that we can use as tumor-specific markers? One then has to look for single base pair mutations or limited insertions and deletions to develop a leukemic-specific profile. So we've demonstrated the feasibility of doing this using a small molecule molecular inversion probe technology called SMIPs. The basic idea here is that one can use a probe that binds to regions flanking the gene of interest. This particular probe has a molecular tag that's unique for that particular molecule. One can then amplify across the locus of interest, open up that construct, further index label this in terms of sample, and what one then develops is a series of molecules that have been amplified by PCR and subsequently sequenced, where the random error associated with the PCR process itself, which is what you run up against for doing these types of assays, can be distinguished from the consistent presence of a mutation across a whole family of molecules as grouped by the molecule-specific identifier that was used at the time the PCR was performed. So this allows us to account for essentially amplification error in PCR assays and distinguish it from the types of molecular abnormalities that we're trying to detect. So as proof of principle, we applied this to AML samples containing the NPM1 mutation. This is a mutation, as we've seen, in about a third of all AML cases. The primary mutation is called type A, and it's seen in about 80% of the cases, but there are about 30 other types of 4 base pair insertions or deletions that occur as well for this particular type of AML. It's thought to be a relatively early mutation or founding mutation, and therefore appears to be relatively stable after therapy for this disease, and that's extremely important for monitoring purposes. And other groups have shown using standard RQ-PCR-based methods that this is associated with outcome. So it's a very good model system to work out these kinds of issues. So doing dilutional experiments, we can show that next-generation sequencing is capable of detecting this mutation reliably and reproducibly down to 1 cell in about 100,000, which is lower than our current flow cytometric assays can detect. And in comparison it with flow cytometric assays, there are subsets of samples where flow cytometry is negative that have residual disease detectable by this next-generation technique. And so similar to what we've seen in B and T lineage ALL, we think that next-generation sequencing is both feasible for MRD detection and is probably more sensitive than the current types of flow cytometric-based approaches that the lab has used up to this point. Then one has to think about the future in terms of how one can employ these technologies to understand the biology of acute leukemia and the way that its genetic heterogeneity exists prior to therapy as well as subsequent to therapy. And so the current techniques that I've been describing are essentially techniques that look at bulk populations of cells, where we're looking at all the leukemic blasts having a similar kind of single genetic signature that we can identify and use for monitoring. But data is beginning to emerge both in B-ALL as well as in AML that that's an overly simplistic view of leukemia. So for instance, this recently published study from the Children's Oncology Group in B lineage ALL was able to show that if you compare diagnosis and relapse samples in B lineage ALL, one can establish various subgroups of disease or subclones within the leukemic population at the time of diagnosis. This is modeled here in this particular figure, part B. 97% of the population appears to contain these 2 particular mutations, but 1% additional of these cells have gained an additional 3rd mutation, another 1% gained a different 3rd mutation, and another 2% have another mutation. So they have some mutational similarities, but also differences that results in some heterogeneity within the population at the time of diagnosis. Therapy is then administered, and in the relapse samples, some similarities are observed in terms of the mutational profile, but some of these subpopulations of cells are lost, some of them are expanded, some of those then acquire additional mutations. So for instance, the most prominent populations of cells here that form 98% or so of the tumor are eradicated by therapy. So there's been an excellent response to therapy on the whole. However, this subset of cells that represented 2% of the sample persists and expands. It acquires additional abnormalities and then is resulting in relapse with a similarly heterogeneous type of molecular profile. So there is heterogeneity in B lineage ALL at the time of diagnosis as well as after therapy, and the relationship between the two differs from case to case. In this particular study, they looked at 20 different pairs of relapse and diagnostic samples, and all of them have different kinds of clonal substructures before and after therapy. The same kind of data is generated in AML. This data came from Washington University, where they did some initial sequencing of AML samples. And one of the important things to recognize about AML, unlike ALL, is that in AML, the leukemic population, although we recognize it as being early progenitors that we call blasts as a representation of disease, those abnormalities actually extend out further into more mature subpopulations of cells. In this case, if one looks at monocytes compared to blasts, one finds very similar mutational profiles between the two, even though we only recognize the blasts or the progenitors as being the leukemic population. Lymphocytes, on the other hand, are rarely involved in this mutational process. One can then do cultured experiments using sorted single cells, such as hematopoietic stem cells, to grow up individual colonies of cells and look at their individual genetic signatures and reconstruct the likely pathways in which these mutations were acquired and show the sequential acquisition of various mutations to form the final frank leukemia, as is demonstrated here by work from the group at Stanford. This can then be used to construct more general models of leukemogenesis, where founder or landscaping mutations involving methylation-associated genes appear to be a relatively early event. Then they are joined by proliferation-associated mutations such as NPM1 or FLT3 or RAS mutations to form a final functional leukemic population of cells that is then representative of what we see at the time of diagnosis. These kinds of simple sequential models, however, may not be entirely representative of all leukemia. And this is data from Jerry Radish's lab here at the Hutch, using relatively simple PCR-based assays on individual single-sorted cells, looking at the presence of wild-type, heterozygous, or homozygous mutations in the NPM1 and FLT3 genes on 6 patients with AML, and showing a very complex heterogeneous substructure within those diseases that really can't easily be described by simple sequential models of mutation acquisition. So there's a lot of activity and ongoing work in these areas trying to define the clonal substructure of leukemia and what's responsible for therapy. But the overall impression that one gets is that this is very complex, and while more work is needed, there's the potential for use of this technology in the monitoring of residual disease. The group at MD Anderson has published some early data of this sort, looking at the presence of multiple lesions in patients with AML, showing, as you might expect, what we've seen already with both B lineage ALL and AML in terms of changes in clonal substructure with therapy. In some patients, such as this first patient, the patient relapsed with the development of a FLT3 mutation that wasn't seen at diagnosis. In this particular patient, there was persistence of a mutation in one of the DNA methylation genes, DNMT3A, that probably is present in a clonal stem cell population that predates the occurrence of leukemia, whereas the actual leukemia itself responded to therapy and was eradicated. So the use of these technologies, particularly in these complex heterogeneous populations of samples, is going to be tricky and requires, I think, a lot more investigation. To the point that perhaps where this field is headed is that what we need to be able to do is develop assays that are capable of detecting the presence of multiple genetic lesions within individual single cells in large populations of leukemic cells in a very rapid manner. And so this really is a technology-driven type of activity, and so this is where the bounty hunter comes in. So in conclusion, what we'd like to say is that classification of these leukemias is largely genetic. Post-therapeutic monitoring is an important, unique prognostic indicator in patients with acute leukemia. It's now widely used in clinical trial development as well as increasingly in clinical practice. Flow cytometry can do this. It's capable of this detection, but there's a lot of heterogeneity in terms of how it's used out in the community. Deep sequencing is feasible and perhaps will be ultimately easier to disseminate as a reproducible technology. We and others have shown that it's generally applicable in B and T lineage ALL. It also appears to be applicable to at least subsets of patients with AML, but probably will require high-level multiplexing to really be made effective for that particular disease. And that in the future, correlated single-cell genetic assignment may be necessary to really assess the possibility of disease relapse. With that, I would like to conclude and acknowledge our staff in the hematopathology laboratory that have been instrumental in the studies that we perform, my mentor Mike Borowitz at Johns Hopkins that I share the Children's Oncology Group reference work with, the entire COG ALL subcommittee that we've worked with now for over a decade, Harlan Robbins and his group at Adaptive Biotechnologies for the next generation sequencing studies, and my collaborators on the next generation sequencing studies, David Wu and Steve Salaponte here in the department. Thank you very much. Are there any questions? Yes?
Do you see any role for using some of these branching DNA probe techniques that you can do with cytometry as well as looking at the phenotype to sort some of this out?
Yeah, so the question is regarding the use of branching DNA technology and flow cytometric assays for this type of detection. The primary use of branching probes that I'm familiar with is an increase in sensitivity to allow one to detect molecules that are expressed at low levels by essentially amplifying signal. That hasn't really been exploited to any significant degree in these types of flow cytometric assays, nor are there commercially available reagents that I'm aware that would allow us to kind of evaluate them. Most of the types of things that we're looking at are relatively larger scale changes in terms of immunophenotype, things on the order of at least a half a log or more in terms of the intensity of expression. So simply amplifying the signal may not improve on our ability to make those kinds of distinctions unless the signal that we're looking at is relatively dim. And most of the folk we've looked at to date have not been in that category. So there might be future for this, but at the moment it's been unexplored. Yeah, Mark.
What a great tour de force. What's known about normal cells or cells in inflammatory states, and how much of this kind of heterogeneity and mutation is part of a normal, or not normal, but found in the adaptive immune response?
Yeah, so the question is about the heterogeneity that one sees in normal populations, perhaps in immune responses or other types of responses. And there certainly is information looking at immune responses using next-generation sequencing types of approaches where one can look at essentially individual cells, or at least cells that have similar types of underlying genetic sequences, and look at relative proportions and changes after therapy and different kinds of inflammatory states. I think the data at that very detailed level is relatively new, but there is some data, for instance, looking at tumor-infiltrating lymphocytes that associates with prognosis and relative expansion or lack thereof of different subsets of cells as part of the immune response to tumor itself. Actually looking at inflammatory states, there I think is a lot of active ongoing investigation using these techniques to do exactly that, but I'm not aware of the actual outcome of that data at this point. That's a good question.
How much genetic information will you need at the single cell level in your made-up world?
Yeah, so the question is how much genetic information will you need at the single cell level to do this type of work? And I think it depends on the kinds of markers that one's talking about and their underlying biology as to what their importance is. So if you were to take markers that are, say, secondary abnormalities that are acquired later during the leukemic process, FLT3 would be one example of such a marker. They're imperfect in terms of residual disease monitoring. They may not be there at diagnosis, they may appear after therapy, they may disappear after therapy. And so if you're looking at markers of that sort, you probably need a relatively larger number of them, 5, 10, maybe even more, in order to fully cover the population heterogeneity. If you go to abnormalities that are present early on, such as founding mutations like NPM1, you probably don't need a lot of additional abnormalities to do that type of monitoring. So I think it depends a little bit on the types of molecular lesions that you're looking at, as well as the particular structure of a particular leukemia. So if you're looking at for a generalized strategy that could be used on, say, all AML, it's probably going to need to be a substantial number of genes that would have to be involved to do this. But the exact number, I think, is anyone's guess at this point. And that's partly what we'd like to further investigate, is exactly what those assays should look like in terms of the degree of complexity. Other questions? If not, thank you very much.